Me, I have some housekeeping here. So we get started. Slideshow, play from start. Okay, my name's Jay Herson, Biostatistics Department at Johns Hopkins. And I'm going to talk to you about the important topic of code review, computer program code review. And always good ships. Now, what does always good ships mean? How would it get into a talk like this? Well, I will explain that shortly. So as an overview, we're going to talk about the retraction. This is a retraction of a paper due to coding errors, a paper submitted by Johns Hopkins physicians to a prestigious journal. And ALA, that was the company that I founded in 1983, and it still exists, although I sold it to Westat in 2001. Always good ships, it comes up again. I'm going to explain that. What were the software applications of ALA? And in 1995, at ALA, what was the motivation for standardization and code review? We'll look at the software development lifecycle and the ALA code review best practices. And then what makes sense for QS, the Quantitative Sciences unit at the Johns Hopkins Cancer Center. What is the retraction? This happened in 2018. Hanan Abu-Matar is a physician in the Johns Hopkins Medical School. She works in chronic obstructive pulmonary disease, a big community. And the biostatistics department at Johns Hopkins has a biostatistics consulting center. Now, at this center, we have many students working several hours a week, coming in, helping with programming, writing program. One student begins a program, another student finishes it, if it has to be divided up like that. And some students are there to pay off, work sufficient hours to pay off their student loans. None of that is particularly troubling, but what happened one day is a student had a program written in SAS, and it was written so that the treatment code of 1 was control group and 2 was the treatment group. Another student came in and was finishing, and he said, well, I don't like this treatment like this way. I want them reversed. So although they were reversed, nevertheless, the paper was submitted to JAMA with the conclusion that the experimental treatment was better than control, which was standard of care. Reasonable results, that's what the grant said they would find. And lo and behold, after publication, it was found that actually, because of that reversal of treatment code, but not reversal of text, that actually the control group was better than this newfangled treatment that they were experimenting with. And so a retraction was needed. And then Dr. Abu-Matar became a born-again Christian and wrote this paper in Contemporary Clinical Trials Communications, which is an open source arm of the journal Contemporary Clinical Trials. And in it, she says, lessons learned. At Johns Hopkins, the error occurred, the retraction had to be made, so the Biostatistics Consulting Center people and the Hannah Abu Matar and her co-authors, let's have coffee and enumerate the lessons learned. If this happened in industry, the lawyers take over, and the lawyers will fight it out, and it goes into the courts, uh, lawsuits, very ugly. However, at least in industry, there are consequences, and in academia, not always the case, except for the embarrassment of a retraction. And of course, the COPD being a big community, many people read that paper originally and said, oh, good, yeah, this way of organizing the COPD treatment is better. Let's use it, and would be unaware of the retraction. Now, the lesson learned that Dr. Abu Matar mentions is check the treatment code at the beginning and the end of the trial and see that they agree. Well, this is grossly inadequate. Why not check all of the variables in the code? Why just treatment? Because that happened to lead to retraction. Also, she is trying to create some notion of quality at the end of the trial, whereas quality has to be built in through all of the phases of the trial, as I will show you. Now, in industry, there's a pharmacy check after unblinding, regardless of treatment codes and randomization assignment. This has been a standard operating procedure across all sponsors in industry since about 1970. And what they do is everyone is unmasked at this point, and trial is over, and we go to the pharmacy. Okay, patient 2101 was supposed to be on the experimental treatment. Mr. Pharmacist or Ms. Pharmacist, what treatment did the patient get? And we go down the list and make sure that it all agrees with the randomization assignment. Now, in a case where all of the controls were labeled as experimental and vice versa, that would not have been a problem with a pharmacy check, because they still have the two treatment groups separated, and now we know what they are. This is not done in academic research, at least not as often as it could be, because it's such a simple matter, and I wish it would be done. Another thing, last year I was serving on a data monitoring committee for a cardiovascular device clinical trial. They made a submission to FDA, and FDA, the results, by the way, were very good. I should mention the primary endpoint was statistically significant, and most of the secondary endpoints were favorably statistically significant. FDA got the results, and they write their own program, not for everything, but they write their own program. And lo and behold, the chi-square test, which was used for all of these statistically significant differences, was in error. FDA said, well, we don't know where the error is. We can't reproduce your results. We're getting different p-values than you are. But it's not our job to find it, so you find it. And then when the sponsor looked, they found the chi-square statistic in the program had an error in the denominator. Now, you know, those of you familiar with chi-square, the chi-square is always observed minus expected value, the denominator square root of the variance. And we might argue, how should we compute the expected value? How should we compute the variance? But nevertheless, that is the form. Well, what was in the denominator of their chi-square statistic, we still don't know where it came from, but it was in no way a variance. The correction changed the results. The primary endpoint was no longer statistically significant, and only maybe one of the secondary variables was statistically significant. And of course, this had to be explained to investors in the company and executives, and something that should have been avoided. Applied Logic Associates was ALA, company I founded, and took the name Applied Logic because both medicine and statistics are examples of applied logic, or so I thought. We had this mantra: we shall build good ships here, at a profit if we can, at a loss if we must, but always good ships. This is a quote from Collis Potter Huntington, who was the founder of the Newport News Shipbuilding Company, a company that still exists today, and we use ships to mean product. And we were always going around the office saying, remember, we build good ships here. And the Huntington family, by the way, in the 20th century, also went on to start the A&P supermarket chain. Our applications, I'm going to go through them, but one was data management. All of our work was clinical trials. They do data entry and database management, and they do edit checks. Edit checks are standard program we run, where the gender has to be 1 or 2, and the blood pressure cannot be greater than 300, and things like that as we learn. And these are edit checks that would apply to all trials. We have specialized ones that would apply only to the current trial. And we have then a database lock when everyone is satisfied that, I should mention, for edit checks, we also have conditional checks. That is, if gender equal male, then pregnancy equal blank, something like that. Database lock is when all of the errors have come out, all corrections made, and it's at that point the data are turned over to the statistical programmers. The software we used in data management was Paradox and Oracle. And we should mention the sponsor does the CRA, Clinical Research Associate site monitoring. They send the armies of people out to the clinical sites to check that the data in the case report form is identical to the data or agrees with the data in the source document. SAS programming then in the data get to the biostatistical group, again, all clinical trials. Data analysis is done by statistical programmers. These are people who have at least a master's degree in statistics or biostatistics. And they know a chi-square when they see it, and I'll explain that as we get more into this. The production programmers, we call SAS programmers, they're making the tables, listings, and graphs. These are output that will be used by data monitoring committees and for the submission to the FDA, and by people at the sponsor who need to write reports about safety and things like that. Of course, we do produce graphs in data analysis, Kaplan-Meier graphs that are part of our data analysis as well. Then we had IVRS telephone randomization. An error in randomization would be a disaster, and these errors happened in other CROs, but not with us. This programming was in C++. A separate unit did this, and they had their own code review, which I will not go into today. I'm going to talk about the SAS programming of the clinical data. So let's go back to 1995. I'll tell you why in a moment. This is a picture of a 1995 Toyota Camry. This car is not for sale, but it gives you some glimpse of the era. President was Bill Clinton. Do any of you out there know what movie won the Oscar for Best Movie in 1995? I'll give you time to think. It was Forrest Gump. Did you see that movie? Well, now that I've brought you back in time, let me explain that we were getting concerned about inefficiencies and duplication. Lots of duplication. Every time we started a new trial, there are new codes for the different variables. And inefficiencies, yes, we were finding errors in the programs before an embarrassing error got out to a client, but nevertheless, this is reprogramming and causing inefficiency. So these are among the sources of error. A lot of it was the lack of standardization of variables. And of course, we had turnover of staff. So someone comes in, says, okay, you're taking over this project. This other person left. And the person looks at it and says, well, I can't read this. I can't make any sense of it. I've got to start all over. And indeed, the person started all over. And this is where there was so much inefficiency. So how would we deal with all this? At about the same time, FDA issued 21 CFR 11 in all of its bureaucratic glory. What that said was people were submitting electronic documents to FDA for review, like a new drug application, and how to make an electronic signature in it. That was maybe 2 pages. It goes on for another 50 pages that sponsors have to validate their programs, make sure there are no errors or minimum errors before the submission to FDA. But they didn't say how to do this, and nobody knew how at that point. Then there was CDISC. This was an industry FDA initiative to standardize the variable names, to standardize other types of things across all sponsors, all indications, clinical trials. And that was a good idea, but we were thinking this isn't going to happen so soon because each sponsor in industry is going to say, I'm in favor of this as long as the industry adopts my codes. And in fact, it took 15 years before CDISC came about. And besides the long negotiations for people to change their ways of doing things, they added new requirements for CDISC. So today, CDISC is a major thing with coming from the protocol, the SAP, it's standardization everywhere and creating a lot of efficiency. However, we created our own efficiency because CDISC didn't exist at that time. I might mention that ALA still exists today as the Houston, Texas office of Westat, and our statistical programmers there who were with us in 1995 are making CDISC conversions for different industry clients. Anyway, we got involved with a client with a cardiovascular device different from the one I talked about before. And TIMI was a series of trials that were coordinated at Harvard and the Brigham. They had a grant, and they gave out money to different companies, sponsors to do trials, and then the legacy data was at Harvard. But anyway, we were to use from a particular TIMI trial, use the data to design a new clinical trial for that same sponsor. And however, for some reasons that I don't recall, the whole clinical research staff of that sponsor had left. They were going to hire new people. Our only contact was an engineer at the company who worked on the design of the device, but not at all on clinical trials. So I wrote to him, can you give us your code book? Give us what are the codes for all, what are the different variable names and what do they stand for is what I mean by the code book. And he didn't answer. 3 emails, phone calls, didn't answer. Finally, I got in touch with him and said, can you give us the book? And he said, but don't you already have the book? I said, no, we weren't given a book. He said, don't all the companies use the same book for these codes? We said, no. Well, he said, I'm an electrical engineer, and the IEEE had standardized everything in 1900. What are you people doing in 1995? I said, well, we're asking the same question. So we said, all right, he got someone to help us on that, but we said, we've got to get busy and we've got to standardize. So data management, statistics, IVRS, all sponsors, variable names, WBC, ALT, AST, that is the same. And in fact, the data management staff would get the blank case report forms and they would write in at the start of a trial, white blood count, there's a space for the number, but they would write WBC, the same for ALT and treatment, etc. And then photocopies of that would be used everywhere. And sometimes we had to create new codes, of course. We would have TRT for treatment, date, treatment start, date last, treatment visit, and CENS for the sensor code. So we then have derived variables, OS and PFS. If we're overall survival, progression-free survival. The formula you see for overall survival is the same in every program because we're using the same variable names that go into it. And so each we walk around and know that formula in our sleep, but we take it off of an inventory and paste it in place, and I'll show you that in a moment. Here is a picture of our data management and stat staff creating standards. And they, among themselves, 2 of these people are from data management, 2 are statistical programmers, and none of them were supervisors. It was a task force we created, but other from the lower ranks, those people know these things better than the supervisors. And all 4 of these people, I might add, have gone on to very responsible jobs in the clinical trials industry. So let's look at the software development life cycle. And this is built into our SOPs, our standard operating procedures. First, requirements. Design and development of the software, testing of it, implementation, maintenance, and retirement. The requirements are given by the protocol written by the sponsor. Now we start with that. Then the design is in the Statistical Analysis Plan, which is it starts with the protocol, but we put our own things into it, like proc univariate is always something good to start with. That isn't going to be in the protocol, but we each line of the protocol has a line number. We now write a statistical analysis plan using our standard variable names, our standard equations, like for OS, where they are appropriate, and those will appear in the statistical analysis plan. So every analytic requirement from proc univariate to the secondary endpoints safety analysis, whichever comes last, is in that, and it's keyed to line numbers in the protocol. The statistical analysis plan also has line numbers, and I'm going to refer to that later, all written using the standard variable names and the SAS statements, and many of them come out of an inventory, as I'm going to describe that in another slide or two. Now let's go back to the chi-square, where the company had a CRO for a sponsor, had that notorious error in the chi-square statement. Our code will have, the chi-square statement will always have OBS minus EXP divided by square root VAR. Now, before the chi-square statement, it's going to be, there'll be a comment that says chi-square. After that, OBS equal, whatever the observation is called in this program. EXP equal, maybe there's a formula after the equal sign for the expected value. And then the denominator is always square root VAR. And again, VAR likely would be defined before the chi-square statement. So you see, we do that, and then we take the same chi-square statement for every program that we write. So the error that occurred with that sponsor could not possibly happen. And as I said earlier, every one of our programs knows chi-square is observed minus expected over square root of variance. And then the whole thing is squared. So, okay, so our chi-square statement then would be chi equal observed minus expected over square root variance, and then CHISQ equal chi times chi. Now, we write the SAS programs with uniform comments. The comments follow a uniform format, and the comments reference line numbers in the SAP. So if I have to compute something, it'll say, the comment will say, computing tumor burden, and line number 129 of the SAP. And many statements are taken from inventory due to the standard variable names. So we have an inventory file that will have the OS formula in it, as I said before. And this was much easier to do when Windows gave us the copy and paste capability. And the copy and paste capability might have existed in 1995. I don't remember that. Now we come to testing. Another programmer reads the code and the SAP to determine if correct and signs off. During the new programmer training phase, we deliberately plant errors in the program to see if the tester needs more training. The more subtle the error, the better. So we might have, so we're supposed to be analyzing ALT and we'll put AST in instead. Or maybe it's supposed to be less than or equal to in some formula, and we'll just have strictly less than. In some cases, and we do destructive testing, entering dummy data, extreme values, to see if error would show up as we would want it to. Also, sometimes we give the programmer in training a program that has no errors in it, to see if they say, well, gee whiz, I've got to find an error. Maybe it's this one. And all of that, we get a lot of laughs at that, but everybody learned very well and accepted this kind of training. I might also mention in the testing, a programmer reading the code might say, gee, this formula for variance, I know it's on line such and such in the SAP, but it doesn't look right to me. Calls over a supervisor. So Joe calls on Susan and says, Susan, this doesn't look right, or can you confirm? And after Susan looks at it, it's right. Whether it was right before or after doesn't matter. It's right. But then a comment is put in place, approved by Susan, a certain date, whatever the date was. So nobody has to worry anymore about that particular program. This isn't rocket science, and it isn't always exciting, but it's something that has to be done. Run the program and correct all errors. That's the implementation phase. And now we can show the results to the statisticians who are interested in what was statistically significant and what wasn't, and what are the safety issues that might have to be faced. Then in maintenance, all changes are detailed in the program cycle database, the program life cycle database, as we go through all of this. And then there's a production qualifier. Another programmer checks how changes affect the output and also signs off. What we mean by that is there may have been a data unlock because, database unlock because we found an error that had to be changed. And then when they run again, did that change the results? And different things like that. I don't remember everything that person did, but we did have such a person. And then the retirement of the program life cycle database is updated for the final history, conforms with the trial closeout SOPs that we had, and we update the SAP programs with the new line numbers, make sure, of course, we've made changes to the SAP and we made changes to the programs, but given that we are now putting the final program away and stored, we want to make sure that if 5 years from now someone comes back and reads that, that they can still have the connection with line numbers. Of course, there will be audits by the sponsor and the FDA, and we're prepared, and they complimented us on this scheme. There are errors of commission and omission. We should just mention commission are the careless errors like we've already described. But for omission, supposing there are two random effects, but only one is put in the statement for the PROC for the Cox's model, or for analysis of variance. There was supposed to be an interaction term in the model statement, and it was left out. The system that I've just described finds not only the errors of commission, but omission as well. The biggest problem is if we meant to have an interaction term, but we never put it into the SOP, that is something that we usually find later, but sometimes harder to find the errors of omission for that reason than the errors of commission. And the same process was done for the data management activities, all of their error checking, and the people that work on tables, listings, and graphs. Of course, data management, tables, and data analysis, every patient has to be accounted for. If there are 503 patients on this clinical trial and 250 in treatment A and 253 in treatment B, then every analysis, every table has to have all those numbers accounted for. So, what makes sense for the QS unit? Well, employee selection, they should have the right attitude. They should be people who are willing and want to be trained and do their programming in this way. And they have to be aware of the consequences for an error. I might mention that at ALA, after we made these changes in 1995, we changed the job description of the programmers that said the programmer spends time doing code review of programs that they didn't write. That's all part of the job of a programmer. And they agreed to it, but we didn't want anybody to be surprised and think they would only be doing programming, which is the way it's done in other companies. But then back to QS again, balance the probability of error, the severity of the error, and the consequences of error. Is the program output for local consumption? Supposing an ophthalmologist comes in and says, can you go back and let me know the different stages of non-small cell lung cancer in a recent trial? Is the visual acuity different by stage of disease? That is not as important perhaps as a final analysis on its way to the FDA, the JCO or JAMA. I think we will agree with that. So again, maybe QS doesn't have to do all of this, but certainly something should be done. Standardization of variables and the program format, definitely. Lots and lots of comments. SAP and the resulting programs are really one integrated system. They are not two separate products. They are one. And review by another staff member, of course, makes a lot of sense. Some closing wisdom. Yes, we are close to the end of this. David Beyer was an MD, but also a brilliant statistician at the National Cancer Institute. And those of you who work in QS may know Steve Piantadosi. David Beyer was a mentor of Steve Piantadosi, and David had a lot of MDs who were statistical programmers. Mitchell Gale would be another one. Working for him. Though I'm not an MD and I wasn't at the National Cancer Institute, I also consider Dave Beyer to be a mentor of mine. And Steve Piantadosi, after Dave Beyer died, wrote a paper about his wisdom and the Dave Beyerisms. The one I liked best was, never trust the work of a person who works alone. And I think that code review is all about that. And remember, whatever you do, always good ships. Thank you.